So we're going to transition a little bit and talk, kind of end with this before you break out into your groups, your subgroups around disease, and talk about some research opportunities. You've heard Dr. Hamvis and Dr. Nogi talk about genetics research and some of the things they're doing. We're going to talk about some other areas too. I'm going to talk about something that we're involved in. Anne asked me to talk about this thing called proteomics, and I'll describe that in a second. And I'm excited about that. And then we're going to hear from our first ATS grant awardee after me, and I will be introducing him. And I think you'll agree that some of the work he's doing is just, it's amazing. So we're excited to have you see that. So let me just spend a little bit of time talking about these proteomic studies and how I see this coming about from chILD. And before I do that, I want to recognize the efforts in the network around research. You've been hearing it again and again, and all the network doctors are very involved in describing the clinical aspects, seeing your patients, thinking about the clinical connections with these different problems, and doing what we call phenotyping. That's a fancy word for saying, how does your child look, and how does this disease look? And all these variations you keep telling us about, that's a phenotype. Well, do they have this component? Is there something about their oxygen level? And so we're all involved in that and needing to capture that. You've heard from Dr. Hamvis and Dr. Nogi about the genetics, but there's other people who are doing very basic science work. Lisa Young working with cells in animals, and Jim Haygood working with fibrosis and cells in animals. So we have people doing research on different levels, and I wanted to bring that to you. So let me talk just briefly about this concept of children with interstitial lung disease and proteomics. And proteomics, I'll define it as a study of what the proteins do. And I'll tell you in a little bit why that's important. And so despite really the huge gains we've had in thinking about how to, let's use knee-high as an example, recognize the CT findings and infant PFTs you guys have been hearing about, and really defining that disease clinically, there's problems that still remain. And even though we have surfactant protein C and all these genetic mutations now, there's still a lot of problems with things that we don't know. So there's still diagnostic risks, and there are those of you who don't have a diagnosis. There's a lot of cost involved still in thinking about the diagnosis, even genetic testing. We get calls from doctors around the country. These guys will know their hospital won't pay for it. And so even the concept of genetic testing and the cost of that is an issue. And then granularity. And what I mean by that is, how good is our testing at breaking down the different severity types, or breaking down it into different mutations or different ways that your child responds? Probably not so good. And then prognosis. Are there markers that we can look at around prognosis? We don't understand a lot of how these mechanisms happen, especially in knee-high. And as you all know, we don't have a lot of therapies. We have not had one clinical trial, and still what we do today is what we think might be good. And even what we think might be good hasn't been studied in an organized fashion. So significant gains, don't want to downplay it, long way to go. And so one of the ways I began to think about that is we need to get outside the box and think about a discovery mechanism for how we can begin to understand that. And by discovery, what I mean is we need to think about taking a bunch of different samples from different places and try to discover if there are patterns that fit together. And I'll show you how we're doing that. Because if we can do that, I believe we can help with diagnosis, pathogenesis, which is another way of thinking about the mechanism of disease, and that potentially we can pick out new treatments. And we'll show you about that, both on the patient level and on the individual cell level, and get to maybe new therapies with trials. If you get to clinical trials, you need markers for that. Your kids some of them are so sick that giving a medication won't register a change until maybe a year down the road. We need an intermediate marker to say it's going in the right direction. And so thinking about these proteins, it's not one protein, it's a network of proteins. So that's the new concept. It's not just one, it's a network, and I'll show you that. So that's kind of a backdrop to what we'll talk about. So let's back up and talk about the system. So if you guys are reading, and you guys are internet savvy, I know, people are going to talk about all these different levels of how we think about how a person, how an organism works. And you've heard a lot about the genome or the genes. And that relates to DNA. That's a lot of the genetic work. Do you have a mutation? And that can tell us what can happen. Your genetic makeup defines what can happen. Now, there's this other little area that we learned about in the conference, which is epigenetics, which is a way that actually your genes can be modified a little bit. But all of this kind of defines what can happen for an individual. Well, the DNA has to be transcribed, so there has to be this little messenger, this intermediate messenger, and that's what we call RNA. So I'm taking you guys back to your biology class in high school or in college. And that's a fancy word for transcriptome. So there's a lot of studies where people can take tissue and they can look for these messages, a lot of these messages, and that tells us what genes are being activated at the time, if you will. But then, so that tells us what appears to be happening. But then what you're trying to do is to make a protein. We're made up of proteins. And your body's made up of proteins, the cells are made up of protein, and it's the cells that really, real time, this is what's happening now. And so that's an area right now that I'm interested in studying. And then finally, there's this thing called the metabolome, which is once the proteins have been involved in a process, there are these small molecules, sugars and nucleotides that can break down and can tell us what happened. So in level of kind of where we are today with medicine, genomics have been around the longest, the Human Genome Project, this transcriptomics or the messenger has come along. Proteomics is kind of coming, there's been problems, and the metabolome is the least developed of the areas. But all of these things together give us this phenotype, or how your child acts. And when people talk about this, this is personalized medicine. Because when we know all these things about one person, it's different. And when we know all these things about one person, it's personalized to that person. So even though we're kind of grouping you, even within groups, kids, people are different. And this idea of personalized medicine is not here yet, but it's through these mechanisms that it's a very exciting time that we can get to. So let me just, again, hit the basics. So you have the gene, the DNA, and then it transcribes. So it's like something reading it, and you get this little messenger RNA that ultimately translates this protein that has to fold all about itself. Oops, why did that happen? Oops, okay. Ignore that knee-high part. Okay, not supposed to be there. So in thinking about the surfactant, you have a surfactant mutation. And when you have a surfactant mutation, it makes one gene, makes one protein, but it may fold. But that one protein interfaces with all these other different proteins. That's why it's a system. So that one gene develops one protein that's abnormal, and that abnormal protein interfaces with a whole system of proteins that creates the disease. So it's these proteins that really create health and disease from one gene. Now, conversely, here's where the knee-high comes in. You can go from a gene that you know about and say, oh, look at all these proteins that it's making, or these proteins are abnormal, and there's signals, a group of signals proteins. Or if you take something like a knee-high where you don't have a gene, you can still look at all the proteins that are not right. And we can say, what proteins are not acting right compared to normal controls, and see, so even though you don't have a gene, you can learn a whole lot about knee-high, the same way you did with surfactant and what's maybe impacting disease. And in fact, if you look at all the proteins, it might even suggest what genes might be involved. So you can use this paradigm in different ways to understand disease and health. So why is this important? And I've alluded to this, all these different proteins, because it's real-time function. It's real time what the proteins are doing and how they're interacting with all these other proteins. And the same gene can have multiple different proteins that have multiple different functions. So you really have to know the proteins they're interacting with to look at their function. And then even though this messenger might be really strong, the protein may not be. The protein may not have folded, may not have worked. So unless you look at this side, you don't know if the gene was doing what it was supposed to do. So the protein is really the end product to look at. And drugs don't interface with one protein. Drugs interface with the whole network of proteins. So you give a drug to interface in a system, and it impacts all these other proteins. So the idea that you have to understand what that system is doing. Jim described the cloud, okay? You have to understand, you can't think in just, it was too simple, one thing happens. No, there's a lot that happens. And the exciting part is we can create groups of changes that can be important and we need to think about, just not one. And so really it's this network that we need to understand to potentially understand disease and health. And as I alluded to, if you know what proteins are going haywire, you can predict what systems they're in, and you can start to say, whoa, these genes are in this pathway, and maybe the gene hit for knee high is in this pathway, and suggests back to the great geneticists, these would be areas to look at. Because even in these whole exome sequencings that can sequence the whole gene, you're going to find things. They're going to find things that are not quite right. How do you know which one? And so these things interrelate back and forth. And so we need these markers that can tell us about these diseases, maybe give us an understanding of what's causing the diseases. And then once you understand these pathways of proteins, there are actually drugs that work in these pathways that are available now, today, that can be repurposed and thought about for treatment. It's actually a very exciting time to have these tools. Now, it's been complicated. So why, since this sounds like a really good idea, why isn't it happening? And it's not happening because it's very complicated. So one gene is going to make, so there's about 20,000, little more proteins that can be encoded by genes, and then they get modified, and so the ultimate number of proteins is actually quite high. And we've really lacked good systems for studying it. And the systems that we haven't been able to measure low levels of these proteins. And yet low levels may be all you need to make a difference. It's not just the high levels of proteins. Low levels may still be important in what's happening. So we haven't been able to measure those low levels. The current system's been expensive and time consuming. And then the data that you get, can you imagine measuring thousands and thousands of these proteins and then comparing them to thousands and thousands of protein? So those of you in your family, if your kids are growing up, statistics. You'll have a job forever practically. Don't you guys agree? Because it is complicated and that's where it's headed. So I'm going to talk today about a system that we have that overcomes some of this, which is why I'm excited about it. So really, this is a quote from the National Cancer Institute, but it's one I believe and I think people would say. It's really the interaction between the genes and the protein and how you think about those systems that are important to understand. And I would also add the genes, the protein, and the phenotype, or what the patient's acting like or the child's acting like clinically. So we need to think about all those, and that's personalized medicine. So let's talk about our kids' problems. So, you know, we see these kids and we hear them, and the knee-high kids are crackling and breathing fast. And this is just Megan probably showed you really pretty pictures. Dr. Guha's from Boston, and he actually is studying knee-high, and this is a stain for a knee-high cell right here. And what he's seen is that these knee-high cells, around these knee-high cells, very specialized other cells come. And so if there are more of these specialized cells, maybe it's putting out something into the airway. And then these knee-high cells themselves put out, chemicals that do a lot of things. So why can't we measure those in the fluid that we do on the bronchoscopy? That was a question we asked. We should be able to. There should be something there that defines knee-high. And then if you look at surfactant, this is a biopsy, well, there's a lot of stuff in there. That stuff should have proteins in it that are different and tell us something. So that was our idea. We could say the same thing if you saw my original pictures at the beginning of the day, bronchiolitis obliterans. That airway is scarred and different. There should be proteins in there that tell us something. Not only in the lung, but potentially in the blood. This is a very active area in not only what I'm talking to you about today, but all areas of disease. Cancer, COPD, everybody's thinking about how to think about these biomarkers and drug discovery. So we asked, can we look at knee-high? We started with knee-high, though we have actually a big bank of this fluid. We keep it in Denver. Knee-high surfactant protein mutations. Could we look at it using a new technique that we have access to that overcomes some of the barriers that I just told you about? and I'll tell you a little bit about it, called somomers. It's a different type of ways to find these proteins. Could it work with this fluid? Could we use this new technology for our kids' diseases? And could the proteins we find, could they be specific for knee-high and surfactant? Because if they can, that could be a way to diagnose it, right? A novel way to diagnose it. Or it may be a novel way to follow how your child's doing or compare it to those who have more severe disease to less disease. And then can we take those proteins, if they're there, and distinguish and actually, now this is doing it on its own. Stop that. And actually take those proteins and say what networks are they in so that we can understand what may be causing this disease. So those were our questions. Could we do that? That's what research is asking these questions. So this is it kind of looks funny, but this is, and now I'm not even touching this. Okay. Oh, maybe I'm touching. Okay. So basically what we're doing with this is that these, okay, this is really a protein, and this is the new development. This is a somomer. So it's been designed to bind directly to the protein, specifically to the protein, and it's designed to bind exactly to the right structure. And they've proven it's specific by all these fancy technologies and techniques. And so that little binding goes to the right protein. And then once you do that, you bind this little thing to this protein, and that's the somomer. What happens is they kind of do a bunch of different steps to, at the end, by washing the stuff that doesn't bind away, and then ultimately they take this little somomer off the protein, and now this somomer measures 1-to-1 to the protein you're trying to measure. So it's a 1-to-1 relationship, and you can measure this. The good news is we know how to measure DNA. All those problems we had was with protein. So this is a different way to look at it, getting back to DNA. And what you can do is in one drop of fluid, you can measure 1,000 of these proteins. Now, I think that in 1,000 proteins, there's going to be differences between knee-high, surfactant, BO, and that was our hypothesis. And in 2014, they're going to have 6,000 proteins in one drop of fluid that they can measure. That's pretty powerful. And so we worked with them, and we did a pilot experiment with our bank, our BAL bank. Some of you probably are in it if you've been through Denver and if you've signed those little consents that we ask you for. And what this fluid is when you do a bronchoscopy with these BALs, there's a standard way you have to do it. So you have to put a certain amount of fluid in to get the right kind of results. And then what you send to the lab, you only end up using 1/3 of it. And so you just throw the other 2/3 away. And so we say, hey, we could throw it away, or can we keep it? And everybody says keep it. It's de-identified, so we can understand more. So that's how our bank grew. And we looked at 6 or 5 kids with surfactant. Most of them had SPC. 5 knee-high kids, 5 CF. We have a very big cystic fibrosis bank. And 5 other kids who had our bronchoscopies done more for airway, noisy breathing. So this is a pretty good group, right? So we should be able to answer our questions either yay or nay. And in this study, we looked at 811 proteins to be able to answer our questions. And so the results really are that there are differences in proteins. So if you look at comparing surfactant dysfunction mutations, okay, we had a couple ABCA3 in there, with disease controls, there were 20 different proteins that distinguished this. If you look at knee-high versus disease controls, there were some. And if you looked at knee-high versus all the others, there were 68. And CF, there's a ton of differences. Okay, so the answer is yes, this technique works. Yes, there are different proteins. That's what I'll show you. Now, this is kind of modern art. When we start getting this, I kind of think of this as modern art. But what I'll show you is that this is called a heat map. And all these individual lines right through here, these are all the 68 proteins that are different in knee-high. So every line is a different protein. And the cool part is every one of these lines is a different patient. So it's a way to display a lot of data at once. And so if you're looking along here, there are controls, there are surfactant dysfunction proteins, there's the knee-high group, and there's the CF group, and these patients or children have different patterns for these 68 genes. and we can know what those genes are. Now, this is a small group, but if you start getting more and more numbers, you start to say, now we have distinguishing proteins for knee high, and that gets exciting to do the things we talked about. We can show you the same thing for surfactant. And what's interesting is there was less of these for surfactant than knee high, and you'd expect just the opposite since it had all that goo in there. But the knee high patients, even though there's not a lot we see, they're putting out these chemicals. So it's the same setup. These are the 18 different proteins now, and these are the different patients. And this group is very different. And specifically, if you look here, where yellow is bright, so these proteins are higher than in surfactant than the other groups, and the red is lower. So we can pick up higher proteins or lower proteins. And the interesting part about these proteins here is that these proteins are also seen in adults who have pulmonary fibrosis. Different disease, but the same markers. So we can start to say, if we understand what our kids are doing, can we help them learn? And if they understand, can they help us learn? That becomes very important to look at these processes. Now, I told you we wanted to look at this, what they're doing. And you can take these proteins now, so those 68 proteins and those other proteins, those 18 proteins, or how many we had for CF or surfactant, and then we had more for CF, and we can put them in a program and say, well, what do these proteins do that are so different? And though what they say isn't as important as the grouping, what we can now do is say, well, in these knee-high groups, those proteins that we found were important in all these different molecular functions, starting to give us an idea about what's happening in the disease. And also note, those proteins are doing different things in the cell than the proteins that are specific for surfactant or CF. So now you can start to say, we get new ideas of what causes the disease. Because in knee high, we've done great things. We have no idea what's causing the disease. So this is discovery. You're discovering new things, new proteins, new pathways. And we've just now run a second one where we have more knee high. Instead of 5, we have 22, more surfactant, ABCA3 and CF. And now we're getting data that looks like this down to single proteins. And you can say, if here is the amount of the units we're measuring by, and here are the different disorders that we're measuring by, in this protein right here, the SPC and the ABCA3 are way, way higher than the controls, the CF, and the knee high. So you can start to say this is an important protein in a surfactant dysfunction. And indeed, we think it is. And as I said, it's also a marker in adult pulmonary fibrosis. So you start to discover things and relationships that you can understand differently. And make a model of this and kind of think and break the disease down in ways that before you couldn't do. And so it's all these proteins that make up a network that may be new. And so ultimately, well, so what? What do we want to do with this? So ultimately, how we're thinking about this is, so if we can, in a drop of BAL fluid, bronchoalveolar lavage fluid, or blood, or grind up tissue, in one drop we can look at 1,000 or 6,000 proteins and come up with distinguishing proteins. We might be able to say, oh, that pattern is consistent with the growth abnormality. Oh, no, that pattern is consistent with knee high, or aspiration, or asthma, or surfactant dysfunction. Now, that's a very different diagnostic paradigm than we have today. And this technology will be a lot cheaper, not associated with radiation, and it might also guide us in how we think the child's doing. We're not here yet, but this is the vision for where this is going. And when you say, well, but my child has maybe LIP, well, maybe that pattern looks a little different. Or you say, my child is unknown. We could say, well, what do your proteins look like, most like? And can we put you in a pathway to think about how to treat you, to get more ideas and just say, gosh, we don't know. Because maybe they line up with surfactant and we just haven't found the gene yet. Or maybe you have a combination, or maybe it's really a subtle difference of knee high. So being able to think like this is how we're thinking about our research. And really, here's the new advances, is once we know these proteins I've showed you, we can look at function. What are those proteins doing? To think about what's causing the disease, get new understanding for new treatments and new diagnostics. So really the next steps in this process are, if you take our children, which I can hear next to me, if you can't, I can, having fun, And you take those who have a problem, be it knee high or surfactant or bronchiolitis obliterans, and controls who don't have disease, and you do what we've been doing in Colorado, which is trying to distinguish rules between what proteins are important in these different bronch fluids, serum, and tissue. And then you apply these rules to a new set. Okay, and that's where really the network comes in, because once we establish these markers that we think could be distinguishing or important in prognosis, we need more serum, or blood. We need more BAL to say, indeed, this is right, before we can actually come up with new testing and biomarkers. And so that is kind of where we're headed in this, is if we get markers that we feel good about, really getting more samples through the foundation and the network to really confirm, are these important? And it's a very different way for how we could approach your children in thinking about diagnosis and disease. And for the patients to come who need to be diagnosed, it may be that we'll use this instead of deciding it need a lung biopsy. We're not there yet. Or even a CT. We're not there yet. Or we say, gosh, you better get gene testing, because it may be that this test is a lot less expensive. So that's just, we wanted to give you an idea about some of the research that's coming that could help your kids. And it's a little bit different than what you're used to thinking about. It's not genes, it's a downstream protein. It's not one protein, it's a group of proteins that will help us. And so that's the work we're very excited about, and Anne asked me to present. Any questions before I introduce the next group? So you may see, if this goes well, that we'll put a call out through the sites and through the Family Foundation that we're interested in collecting samples. If that happens, we'll be able to send you a kit and for you all to participate to try and further this research. Any questions? Okay.